Hi, my name is Sarah Taylor and I'm a senior scientist in the applications group at Transcriptomics. I'm here today presenting my poster on analyzing infiltrating B-cell populations in the tumor microenvironment using single-cell transcriptomics. At 10x Genomics, we recently launched our single-cell immune profiling solution, where we can generate gene expression and repertoire sequencing libraries from the same single cells. We use the 10x technology, which partitions cells into droplets with gel beads, and then perform reverse transcription, individually barcoding the single cells so they are uniquely addressable. We then perform cDNA amplification of all the cDNA we generate in the RT reaction, and from that amplified cDNA generate multiple sequencing libraries. We use one aliquot to make a gene expression sequencing library with a standard workflow. And we take another aliquot to specifically amplify the TCR sequences or the IG sequences. And that's done using a targeted enrichment PCR with 2 PCR steps to really allow us to get the high resolution we need to look at these sequences. We sequence these on any Illumina platform and then run through our pipelines we've developed in-house specifically for these analyses. We use Cell Ranger 2.1 and then go into we use our Loop cell browser or our Loop VDJ browser to visualize the data. So we perform this workflow on a colorectal cancer tumor sample. And what we were really trying to do is look at the different cell types that we would see in that cell population. And so if you look up here, you can see when we really try and characterize this environment, we see many, many different cell types within the tumor. We see the epithelial or tumor cells, and then we also see some dead cells characterized by high expression of mitochondrial genes. What's really interesting is that we see a very big immune cell infiltrate. We see T cells that we can subset out into cytotoxic T cells and helper T cells. And we also see B cell populations and mature plasma cells. So the really nice thing about this assay is that in addition to the gene expression information, we get the TCR and the IG sequences. And that tells us a lot about what's going on in the tumor and more comprehensively analyze the infiltrate. And so this figure here is showing histograms of the frequency of the clonotypes that we detect. A is showing the B cells, and what you can see here is that we have one clone that is present at a much higher level than all of the others, around 4% of all clones detected. And so inferring that there's been some B cell clonal expansion. When we look at the T cells, you don't see this. So the top clonotype here is a 1%, and it doesn't particularly stand out. So suggesting that within the T cell population, you don't have a specific expansion. And so what we now are able to do with this technology is link this information together to really identify, so what type of B cell is this? Like, we can look at the gene expression and see the stage of maturity, or is it even a plasma cell? And sure enough, when we intersect this data, as is shown in the figure here, we can really get at this functional phenotype. So this plot is showing the t-SNE plot from above. We took off the color and then overlaid the clonotypes. The small blue dots represent any of the B-cell clonotypes that we identify at all. And the dark blue dots represent the top clonotype that we see in this figure here. And so what we see is that this top clonotype localizes to the plasma cell cluster, suggesting that there's some tumor-specific response being listed within this tumor. And so that's really nice because now we can really get at the functional phenotype, which we haven't been able to do before. So the conclusions from this poster, the workflow allows you to robustly characterize and differentiate the major tumor and non-tumor cells. We get a high-resolution interpretation of the immune repertoire with the paired TCR alpha and beta sequences or the paired heavy and light chains for the B cells. And combining this data for the same single cells allows us to really understand the functional phenotype of the cells within the immune cell infiltrate. So thank you very much for listening to my poster and hope it was helpful.